Hello, today we're going to talk about the Kidd blood group system. The Kidd blood group system was discovered in 1951 when anti-JKA was found in a pregnant woman named Miss Kidd. The null phenotype JKA and JKB negative was discovered in 1957 after a woman had a transfusion reaction and was found to have anti-JK3. The ISPT symbol for the Kidd blood group is JK, and 009 is the numerical designation. The Kidd blood group has three antigens, JKA, JKB, and JK3. The Kidd gene is called human urea transporter 11. It is located on chromosome 18. This gene encodes a urea transport protein that can be found on red cells and kidney tissue. The JKA and JKB alleles are inherited as Mendelian codominant. A third allele, called JK, is a silent gene that produces no gene products. Homozygous JK individuals will phenotype as JKA and JKB negative and can make anti-JK3. A dominant suppressor gene, called INJK, was discovered in a Japanese family in 1986. This gene is independently inherited from the Kidd gene. This gene inhibits Kidd antigen production, making it look like individuals are JKA and JKB negative when really they are not. The antigens can still be seen with absorptions and elutions with anti-JKA or JK3. These individuals cannot make anti-JK3 because they still have some residual Kidd antigens on their red cells. The Kidd gene product is an integral glycoprotein that passes 10 times through the red cell membrane. The fourth extracellular loop carries the JKA and JKB antigens. The third extracellular loop carries a small amount of ABO antigens. Approximately 1% of the total ABO antigens are found on the third extracellular loop. The Kidd glycoprotein is a urea transporter in the red blood cells and kidney tissue. It is important in urea and urine concentration. In the kidneys, the urea concentration is controlled by the Kidd glycoprotein and allows the kidney to maintain a high concentration of urea, which is important for urine concentration. In JKA and JKB negative cells, urea transport can be up to 1,000 times slower, and these individuals often have trouble concentrating their urine. It is also believed that the Kidd glycoprotein plays a role in the cell's osmotic stability. The Kidd antigens are well developed at birth and are found on red blood cells and kidney tissue. They are not present on platelets, lymphocytes, monocytes, and granulocytes. The Kidd antigens are not very immunogenic, meaning they are not very likely to cause an immune response. Only 7 out of 1,000 individuals will form anti-JKA when stimulated with JKA-positive blood. The Kidd antigens are enhanced by enzymes such as physin, papain, and trypsin. They are resistant to DTT, chlorocone diphosphate, and acid. People with the null phenotype lack JKA, JKB, and the JK3 antigen. There are two mechanisms that can result in the JKA negative, JKB negative phenotype. The first is the amorphic. This is the true null phenotype. These individuals are homozygous for the JK allele, which is a silent allele that produces no gene products. These individuals can make anti-JK3. The second mechanism is the dominant suppressor. This is the IN-JKA. These individuals still have residual amounts of JK antigens on their red blood cells and cannot make anti-JK3. Most individuals with this phenotype have been developed in the Polynesian populations, such as the Pacific Island origin, Philippines, Japan, Hawaii, New Zealand, Samoa, and Tonga. Cells with the JKA-JKB negative phenotype are resistant to lysis by 2 molar urea. This can be used as a cheap method to screen for the JKA-JKB negative cells. If cells are placed in 2 molar urea and they do not lyse, then they are likely to be JKA and JKB negative. This table shows the phenotype frequencies for the Kidd antigens in Caucasians, African Americans, and Asians. Caucasians and Asians have relatively similar phenotype frequencies, except that Asians are more likely to be JKA and JKB negative than the other two ethnic groups. This is because of the dominant suppressor, INJK, that is found in this population. African Americans are more likely to be JKB negative than the other two ethnic groups. You should be familiar with the phenotype frequencies of these three ethnic groups, as they often come up in exam questions. KID antibodies are clinically significant and usually comprised of the IgG1 and IgG3 subgroups. Some IgGM can also be present. KID antibodies are best reactive by the anti-human globulin test. Up to 50% of KID antibodies can bind complement. This complement binding usually comes from the small amount of IgGM antibodies that are present. They can also cause in vitro hemolysis. KID antibodies are known to demonstrate dosage. This means they react stronger with homozygous cells than heterozygous cells. KID antibodies are immune stimulated, meaning they arise from outside stimulation from pregnancies or transfusions. KID antibodies are enhanced by enzymes such as physin, papain, and trypsin. They are resistant to DTT, chlorocone diphosphate, and acid. KID antibodies do not store well and fade quickly from circulation, meaning that a patient may have a negative antibody screen even though they have a history of KID antibodies. This is why good patient history is important in blood banking. KID antibodies are known to cause severe hemolytic transfusion reactions and are notorious for causing delayed hemolytic transfusion reactions. Up to 1/3 of all the delayed hemolytic transfusion reactions are caused by KID antibodies. KID antibodies rarely cause hemolytic disease of the fetus and newborn, and when they do, it's usually pretty mild. Since KID antigens are located on kidney tissue, KID antibodies have been implicated in acute kidney transplant rejection. Auto-anti-JKA, JKB, and JK3 have also been found in patients with warm autoimmune hemolytic anemias. Anti-JK3 is only found in JKA negative and JKB negative individuals. It is composed of IgG antibodies and best reactive by the anti-human globulin test. Anti-JK3 will react with all cells that are JKA positive and or JKB positive. Anti-JK3 is known to cause severe delayed hemolytic transfusion reactions and mild hemolytic disease of the fetus and newborn. Compatible blood can best be found by typing siblings of the patient or from the rare donor program. Anti-JK3 is most commonly found in the Polynesian and Finnish populations. In summary, KID antigens are found on the human urea transporter 11 protein, a urea transporter protein located on red blood cells and kidney tissue. KID antibodies are clinically significant, comprised primarily of IgG, and can bind complement. KID antibodies are known to demonstrate dosage, meaning they react stronger with homozygous cells versus heterozygous cells. KID antibodies are notorious for causing delayed hemolytic transfusion reactions and can cause mild hemolytic disease of the fetus and newborn. KID antibodies tend to decrease in titer over time, meaning a patient's antibody screen may be negative. This is why it's important to have good patient history. KID antibodies and antigens will be enhanced by enzymes and resistant to DTT, chlorocone diphosphate, and acid.